In this video, we will discuss about the recruitment of monoclonal antibodies to AND gate logic. So far they have tested the AND gate logic system that it can be specific when both the biomarkers are present on the cell surface, or it can also work for the single biomarkers, and it can do the drug loading also, that they have also tested by checking the cell viabilities by delivering the MMAE drug or the commercial drugs, that is DXT. Now in the final main figure, they have shown whether they can enhance their AND gate logic system by attracting other monoclonal antibodies that can target the tumor cells. So in this case, in order to test their system, first they check it with the different size of the DNA. So when they check it with the hairpin loop, and they also check it for the 21 nucleotide SCR product and 42 nucleotide SCR product, and in this case they have, in this 21 and 42 nucleotide case, they have two places of this fluorescence where fluorescence signals can be formed, and they checked it with this humanized anti-fluorescein antibodies. So for the case where they use hairpin loop, they are able to see very minimum signal or almost no SPR signals that they are able to detect. But when they use the 21 nucleotide and the 42 nucleotide, they are able to see signals for the fluorescence, so that they can calculate the KD and K of value for the SPR. And 42 nucleotides give the better results, slightly better results than the 21 nucleotide. And one of the reasons that they mentioned that this could be because there are more probability available for these fluorescein antibodies to bind to the 21 nucleotide or 42 nucleotide SCR product, because it has two different positions where fluoresceins are available. And when they check it for their actual product that has the multiple, or when they check it for actual product or the multiple SCR, this hybridized chain, not multiple, when they check it for the actual product, that is hybridized chain reactions, since they have multiple sites available for these fluoresceins, binding affinity is almost 10 times higher compared to the 21 nucleotide, that is 3.4 nanomolar compared to the 3.8 nanomolar, and that is evident from the SPR signals. Now they tested that their SCR, they can design their SCR in a way that it can attract monoclonal antibodies. So they tested for their actual system, where what it is, they use the dual biomarkers for this PTK7 and EGFR. At the same time, they modify this SCR in a way that it can attract both this protein G fluorophore and anti-FITC. And in order to see the positive signals, so they also use another antibodies, that is Light 405, which can target protein G. So what it will give only signals only when this it can interact with this protein G and the anti-FITC, which is from the another biomarkers. This is their actual experimental design. First, they will bind this protein G and this, then protein G and this anti-FITC antibodies, and in the second case, what they will use is this Light 405. Light 405 will give signals when it comes and interact when these two antibodies are close to each other. That is, in this case, since these are close to each other, so it will give signals for this 405. And they test it for the different combinations, and they are able to see signals only when the protein G and anti-FITCs are present, indicating their specificities and the ability to recruit monoclonal antibodies. In these things, they have quantified the penalty, which further confirms what they have seen in the immunofluorescence results, that when both these anti-FITC and protein Gs are present, then only they are able to see signals for this Light 405. Otherwise, in other cases, they do not see any signals or almost negligible signals. And one more observation that they have seen, that similar to the case where they used MMAE3 3 times per Aptamer or Affibody, that there is a cluster formation occurs at the cell membrane, indicating that this cluster formations also occurs because of the antibodies are clustered together close to each other, and this could have enhanced this recruitment of these monoclonal antibodies, that is both anti-FITC and protein G. So I hope you understood. So this is how they validate, not understood. So this is how they validate that their AND gate logic based system can also recruit monoclonal antibodies. Thereby, it can also, there is also another dimensions to further increase the efficacies of their drug delivery system to kill or suppress the tumors. Yeah. So I hope you understood how they validate their HCF AND gate logic system for the recruitment of monoclonal antibodies, and how they have also shown that it is specific, very specific, when both the biomarkers are present. It is very specific to the design. That is, when both the biomarkers are present, that design is shown in this figure. Results for that design is shown in this figure. So if you need to understand more about their experiments and wants to refer to the supplemental figures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.